All right, hello everyone. We're back and we are going to have a little fireside chat with Elsa Fouragnan and from the University of Plymouth. So Elsa, why don't you start by telling us a little bit about yourself and your lab?
Yeah, well, thank you very much for having me. So I'm Elsa Franian. I'm a UKRI Future Leader Fellow, which means that I'm 100% neuroscientist. I just do that. I studied the prefrontal cortex mainly, different areas of the prefrontal cortex that are related to decision-making, learning. And it's quite interesting, all those areas, particularly when something goes wrong with it, because then, yeah, it might be linked to different psychiatric disorders, and it might be interesting to better understand how they function, to maybe even help people ultimately. And more recently, I've been working a lot on using transcranial ultrasound stimulation in humans, potentially to better understand certain parts of the brain, but also perhaps to help with real mental health applications. So we're based in the Southwest at Plymouth and at BRIC, the Brain Research Imaging Center, which is the largest of its kind in the Southwest.
Elsa, what time is it over there? It's 10:40 at night or 9:40?
It's super late, yes.
Okay, historically, we've had very good luck with our late night session, so I think this is going to be a ton of fun.
Very much looking forward. Okay.
Yeah, go ahead. Tell us about your work.
Okay, yeah, so well, I'm going to just show you one particular study that we've been doing over the last year or so. And what we were interested in is whether or not it is possible to use TUS in humans, and so TUS is short for transcranial ultrasound stimulation. I use it as my favorite acronym. And whether we could use TUS to modulate the human brain. And what we were interested in is neurochemistry, so the change in certain chemicals in the brain after TUS, and also network change. So how, if you are modulating a certain part of the brain, how does that make this part of the brain communicate differently with others? So network change. So just gonna move to my next slide if I can. And I can't. I don't know why. Yes, I can. Okay, so background study is a little bit twofold, the background. On one hand, there's this brain area that's called the dorsal anterior cingulate cortex, which I'm quite obsessed about. I think it's one of the coolest parts of the brain. So it does a lot of things, the cingulate cortex, but one of the things that seem to be very important is the ability for this brain region to allow us to switch to other alternative and to carry the what if question. What if I stop what I'm currently doing and I do something else? What if I was stopping this relationship or doing another job, or just something that is currently ongoing, having the ability to evaluate that and switch to another alternative. And this is very important for like foraging in animal studies, but also in humans, right? To just be able to change from what we're currently doing. And this in psychiatric conditions is very important because there are a lot of them where people are stuck in certain loops. So you can think about OCD or depression, rumination, addiction, all of this, the ACC is kind of a hub. So there's this part of my background, which is about the functional role of this brain region. And then on the other hand, there was all the work that I was doing when I was still in Oxford on TUS in non-human primates. And what we found is that, so this part of the brain here, if you see my mouse, this is a macaque brain. So it's a lot smaller than a human brain. But this part of the brain here is kind of the analog to the dorsal ACC. It's called the perigenial ACC. And if you apply a 40-second offline TUS, so a 40-second train of pulses to this brain region, you change significantly how it's going to communicate with the rest of the brain for up to 2 hours. So this was really cool when we found that. And then we went further and we checked whether it had a causal role behaviorally. And we found that, yes, indeed, if you perturb this brain region, macaques have a harder time in switching to a better alternative and worse. They became more exploratory. if I want to just make it really short. But what was super interesting is that they were very good at everything else. So it's not like we were completely changing the way they were behaving, but we were making them more exploratory. And particularly when we were looking at switching behavior, we could see that it was very different than a normal behavior.
Was it more like they were doing trial and error than kind of thinking about, what if I did this?
Well, what we think is that they were less likely to allocate the right counterfactual value to counterfactual alternative. So they're supposed to be tracking the value of everything in the world, but somehow they were a bit more myopic. So they seem to be more interested in what they were currently doing and keeping less in mind what they could have done, or you see the alternative things that they're not currently sampling from. They were less able to keep the value of those alternatives. And we could see that quite clearly. So if you cannot evaluate your alternative, well, you just don't remember if it's a good idea to switch.
And what drew you to ultrasound? Because I know your former work, you've sort of experienced all the other non-invasive techniques, but ultrasound hit you in particular for this brain region. What drew you in there?
Well, you're talking about the more traditional brain stimulation, like TMS, TDCS, and so on?
Yeah.
Well, ACC is quite a deep cortical region. So classically, you just can't reach it with TMS. So it's still cortical. So it seems like it's quite superficial, but it's not. In fact, in the human brain, it's like 4 or 5 centimeters deep.
Has anyone ever even been able to try to do this region with TMS, or is this really the first time anyone's been able to non-invasively target this? Which is exciting. I'm trying to get our listeners hyped up. Not be done any other way, maybe.
Well, I think that there is very sophisticated state-of-the-art TMS thing now and you can also perturb network. So you could argue that if you stimulate a certain brain region that's super connected to the ACC, in a way, you're indirectly modulating it. But yet, to my knowledge, TUS is the best tool.
Well, it's interesting. People like all of us at some point have written in a grant or something that, these other techniques are excellent for cortical structures, but this is technically still cortex, and it's really not that easy to get with these techniques. So I think it's important for people to see that even these lower cortical regions, this is the tool. This is the best.
Yeah, exactly. And it's also because it's so specific that you can play around with even, columns of the cortex. Yeah. So I would say that now almost this works against us because, you're used to like a Brodmann area. So the ACC is Brodmann area 24, right? But it's huge. So if you go there with your focal volume that's just 1 millimeter large, it's actually very small. So maybe we would want to have a TUS, a focal volume that would be slightly more enlarged. I don't know. That's
a problem to have.
Yeah. Cool. Do you want me to go ahead?
Yeah.
Okay. So counterfactual value in one hand, and then we know that it is possible in the macaque in the other hand, but can we do it in humans at all? So before we engaged in looking at like the very exciting things, which is to modulate the ACC when people are doing something, we wanted to investigate whether it was possible to just induce any change at all while people were at rest. So we decided to look at 2 brain areas because it's kind of, just if you're thinking about a cool experimental design, one that's robust, you might think that it might be important to have an alternative region to show that your effects are specific to the region that you're targeting. So, if you only have one region and sham, well, somebody could argue maybe you've just perturbed the whole brain and it's not just specific to that brain region. So to counter, your counter argument would be, okay, I will have another active control region, and we'll see if the effects are specific. So we decided to take the posterior cingulate cortex because it's a very rich, high metabolic part of the brain. It's super connected to a lot of sensory regions, but it's also because it's super active at rest. And we wanted to put people in the scanner at rest and just see the effect of TUS on the brain. So yeah, we chose this brain region for this reason, because we know it's highly active at rest.
Elsa, is it, I guess, why is it highly active? Is that part of this cingulate cortex also involved in switching decision-making pathways? Because they're not really making any decisions laying down in the scanner, right?
No, they are mind wandering. So mind wandering is a switch between a bunch of different states, but one of them that's very recurrent is the default mode network. So if you're just mind wandering, somehow, yeah, the PCC, the VMPFC, the ventral medial prefrontal cortex, and other parts of the lateral areas would be activated. So I'm not too sure exactly why, when you're mind wandering, the PCC is so active, because maybe it needs all the representation, the sensory representation, the visual representation. So you have a lot of, if you're mind wandering, maybe you are holding on to certain visual representation and so on, or perceptual representation. So maybe that's why.
Okay. Either way, it was a nice choice, because I think something that's going to come up in season 3, which Kim and I are very excited about, and Elsa's bringing it to the forefront, is that if you want to see things get interrupted and go down, you should probably start with things that are high to begin with. So we support your choice, Elsa.
I think it's also I'm actually going further now for future study, in case somebody wants to investigate this. I think that it's very important part of the brain for different depths of anesthesia, but also different depths of awakeness. So is that a word in English? So if you're sleeping or awake, the PCC will be very differently active. So yeah, just an idea. So we we wanted to use MR readout after offline TUS. So offline TUS means that you're delivering TUS for quite a substantial amount of time compared to when you're doing it concurrently with your readout. So here, the protocol is an 80-second train of pulses, and then we have the readout that can last up to an hour. So the one we were interested in is spectroscopy. So here, this is just a spectrum, and that represents the resonance, the frequency at which certain groups of proton along different molecules are resonating. So you can just have with that the concentrations of certain metabolite in the brain in a certain specific region. So we were mainly interested in Gaba, the main inhibitory neurotransmitter, but also, yeah, it's not just a neurotransmitter, but anyway, of Gaba in the brain. And we also wanted to look at resting state fMRI. So this is just looking at the relationship between brain regions, so how they correlate, they co-vary. So this is like co-variation between a time series of different brain regions. So the idea is that you take your time series in one brain region, and then you look at to what extent it co-vary with other brain regions. And then you have those maps, which has maps of correlation.
Is that other peak glutamate? And why are you more interested in GABA than glutamate, or why not glutamate? Yeah.
Yeah, so I mean, I think we would be equally interested, and even maybe more to glutamate, because so the part of the cortex that we're interested in are mainly excitatory. I've just figured this out. That there is more excitatory neurons than inhibitory neurons. But the sequence that we use, which is called megapress, is really good for GABA. It's edited for GABA particularly. So it will have a reliable measure of GABA, whereas glutamate is not going to be as reliable. So it was both pragmatic, but also of interest. Yeah.
For people looking to do these studies, from that review that Elsa made Kim and I read before the talk, which we're really excited to do, it's something that's easy to do alongside of fMRI, right? So you can do the GABA measurements and you can also do blood flow, sort of not simultaneously, but quick, or like how easy is that? Did you do fMRI while you did this?
I think it's a misconception to think that you can just squeeze in an MRS voxel and get good measurement, because it takes quite a while if you want a small volume. So we wanted smaller volumes because, well, smaller than the normal. The normal volume, I think, is 3 by 3 by 3, which is pretty big. It's a huge chunk of the cortex. But because
millimeters or
no, centimeters, I think.
Very big.
So that's 2 by 2 by 2.
Yeah, that would be centimeters. Yeah.
So
Wow.
Definitely centimeters. So yeah, it's huge, and it feels like the TUS focal volume will be like washed out within a big fat MRS voxel. So we tried to keep it super small, but the SNR is, I think, it's proportional to the square root of the number of volume you acquire. So say you want to reduce your volume by half, that means you have to collect 4 times more in time, right? 4 times more averages. So for that little volume there, it still takes almost 9 minutes.
So the SNR is going to go linearly with voxel volume, but then to regain that signal to noise back, you have to have twice the scan time. Yeah,
exactly.
Yeah.
4 times. So it's a cost.
Yeah.
I think originally I thought it was naively, I thought, oh, we'll just squeeze an MRS voxel here and there and there. No, you have to like optimize things. So we had, I'll show you this on the next slide, actually. I'll show you exactly how we did this. Okay, so this was all collected and analyzed by Siti Yaku, who is a postdoctoral research fellow in my lab. So yeah, 24 participants came. They first came for an MRI, just a T1 weighted image. And we used it both for neural navigation. So when we are doing the TUS, when we are applying the neuromodulation, but also to get information about the bone. So this is a deep learning method that she's developed, where from the T1 we can get bone information, and from that bone information we can have some relatively reliable acoustic simulation. And that's important for safety, but also for efficiency. You just want to make sure that something is going through, something that makes some sense, and it's where you think it should be. So that's what we did with the first MRI scanner that we were getting from a participant. And then they were coming 3 more times. So they were coming 3 more times, and I will describe what happened for each time. So these 3 more times happened at 3 consecutive weeks, where they came at the same day of the week and same time of the day. That's because Gaba is fluctuating during the day, so we were trying to minimize risk of just natural variation. So they came, we made participant MR safe. So we removed, all metal and everything, made them all good and ready. Then we did neural navigation. So this is when you put, your little tracker on your transducer, and then you navigate with the T1 of the participants where you want to go. And then you just, hold the transducer for 80 seconds and you apply your protocol. So the protocol that we used, the credit is going to Robert Chen's lab here. It's in their lab that they developed this protocol. It's called a theta burst TUS protocol. So it has a theta pattern TUS. So 5 repetitions every second. And the duty cycle is 10%. And it lasts 80 seconds. We can talk about the protocol if you're interested. But what I know is that in their study, when they were looking at the motor cortex, it's with this protocol that they actually gained the highest change in MEP, motor evoked potential, compared to other repeated TUS protocol. So we used that. When they were not receiving TUS, we played a sham sound. So they had this bone conductive headphone that were just put in their head. And we always put them in, so they couldn't really tell whether we were just playing the sound through the bone conductive headphone or if they were receiving the TUS. And if you want to hear, this is how it sounds. Can you hear it?
Let me click it again.
Oh, it didn't work? That's a shame.
I think maybe it sounded like your mouse clicking, but it was actually the sham.
No, I think I need to share computer sound. There you go.
Oh, yeah.
Yeah. Can you hear that?
Yeah.
Yeah, that makes a lot of sense for that 20 millisecond sound to sound like a yeah, it's a very complex frequency components. One of these days I'm gonna have to talk about the auditory stuff.
Yeah.
Let's not digress on that. Let you keep going.
But there was one thing I needed to tell you guys, because I know you're diving, right? So you're used to snorkeling, and they have created this amazing snorkel that has a radio receiver and it conducts through the teeth.
Oh, how cool is that?
How cool is that, right?
Yeah, you can talk to
want-
each other or something, or listen at least. Listen to the radio while you're diving.
It must be cool if you're just swimming and just, well, Anyway.
You already talked about your two brain regions though, right, Elsa? Because when we were at the Fun 22 conference, we were all looking out for it. And one thing that was awesome about your study is that you had a sham, but you also have another brain region where they're going to hear pretty much the same thing. You're moving the transducer kind of slightly. So if those have different effects, the sham doesn't matter as much, right? Some people think shams are no good. I think it's awesome, by the way. When I heard it just now, really awesome. But
I'm confused by your study because you say sham and active TUS, but wouldn't you have to do an MRS study after the sham as well as after the active TUS? Did you do 2 MRS studies on each of these, on each session?
Yeah, we have 2 MRS. So let me show you that. We put then people in the scanner, right? As quick as we possibly could. So this usually like 2 minutes. Yeah, you still need to make sure that, they're not running and flying. So once they're in, we had to actually acquire another T1. So that was an extra, 2 minutes, 3 minutes to place our MRS voxel. But so we acquired bold for like 5-6 minutes. Then the first region, the second region, and then another resting state fMRI series, which was quite cool because that means that we can have a look at the network just 15 minutes after, but also 45 minutes after. And these 2 brain regions were counterbalanced, right? So sometimes it was the ACC, sometimes it was the PCC first, so that there was no effect of time.
Yeah, but I guess my question was, for each of the sessions, did you do sham and active and do an MRS after each of them, or was it sham or active TUS?
Or.
Oh, I see. Okay.
Yeah. It's just that we wanted to at least keep the sound. Everything quite. I've been thinking a lot about sham, and it's a tricky thing to do. It's not like you can. Well, even if you were to sonicate in the air, we can't do this obviously, but then they wouldn't hear the sound, and that would be problematic. So you still want them to hear something. So that's the best we could come up with. Maybe the best way would be actually to sonicate, but have all the beam going in all the directions so that nothing is, focused to a specific point in space. Maybe that would be a perfect sham, but then you would still be doing something to the brain. So don't know, but yes. We don't think that it has too much of an effect though, because, remember the different, the time between the sonication period and the first acquisition is you have like 15 minutes in between the two. So I don't really think that there would be some confound there. I'm happy to be challenged on this one, but I don't think it's a problem.
Well, you had the active control, which is like gold standard. So you could have probably have not done the sham at all. And would anyone have complained? Kim, would you have complained? Imagine no sham at all, just 2 brain regions. They should sound the same. I think it's.
I mean, I think the active control is better. I mean, I think there's still some question is, does it sound exactly the same? But, you know, it's probably reasonably good and better. Yeah. Mm-hmm.
Yeah, I still think that it's quite cool to have the sham because you have your, I don't like the word baseline, but you see what I mean? You know how things would be in a total normal sort of circumstances. So you can still look at variation across people maybe, and there are things that could be important from the sham.
You guys are going to see me outside the next conference with a big sign, no more sham. I just like the active control. That's what you got them both, Elsa.
Yeah.
We're happy.
I mean, we're really.
I think you make a good point that, you know, sonicating some other part of the brain, maybe that does something. So, you know, I guess if you do the active control and then you say you have the same result as the sham, that would be the most strong.
Yeah. Oh, that's true. That's a good point.
Exactly. Yeah.
Let's see what that sham did, Elsa.
Sorry?
Let's see what that sham did in the
next slide. Okay.
me and Kim are holding you back here. We're addicted to this auditory stuff.
So this is just an example. It's kind of a good example. Some of them were maybe not as nice, but yeah, I wanted to just show you kind of the evolution from the T1 to the pseudo CT. So that's, you know, trying to replicate Huntsfield units from this image, and then what you can get from the simulation. So you have your acoustic field, and from the acoustic field you can get your full width half max, and you can just threshold at this point and take the maximum, the volume, the focal volume. And this is what we did. And then we, so you cannot really see it here, but there's 3 MRS voxel on top of one another for the time when we were sonicating the sham and the DACC. So obviously, you know, there was sometimes a tiny bit of variation because we had to put them manually in there. And but overall, we were quite happy. And I'll show you a tiny little video here of so the map here is the overlap between all the focal volumes across participants. And you can see here the individual participants' focal volumes. Just for the PCC, actually, I could have shown you for the ACC as well, but I think that makes a point. Yeah, I think this video is really cool.
It's awesome. It's amazing how small it is. And what kind of transducer is that? It's a 500 kilohertz or something, or?
Yeah, it's a 500 kilohertz. Yeah.
Yeah, it's spectacularly small. And if you plot intensity, it looks even smaller. That's a fun little trick for publication.
Yeah. And this is the results of the simulation. So I think from it, what is important maybe to check certain, parameters and make sure that you are safe. So this is the in situ values, right? When we account for the transmission through the skull. And other things that are quite maybe interesting is the fact that I'll go back to my previous slides. Maybe it's more clear on this one. Because the PCC on average is deeper than the ACC, maybe by like a centimeter, sometimes less, but you still have a more elongated volume. So basically, when you're thinking about target engagement, it starts to become an issue to actually sonicate different parts of the brain because you're shooting maybe more tissue with the PCC than the ACC. That's what I'm trying to say here. So let's keep this in mind. Right.
So you're well within the FDA regulatory limits on the intensity and the MI. So
yeah. Right, yeah. Okay, so let's now look at the main slide. After that, it will be very almost quickly the end. Right. So I'm showing two two-step in the way we were looking at the results. And the first is because we didn't really know our effect size to start with. We pre-registered the study and we said at N15, we're going to have a look at the data. And then we're going to decide how many more we need because we will be able to estimate our effect size. So that's why I'm showing you effect with two different sample size. But what's very clear, so this was the results for the PCC voxel, so this brain region here, and the GABA concentration. So when I say the active control, it means that we're looking at the GABA concentration within this voxel, but when the ACC was stimulated. Then when there is no stimulation, that's sham, and after stimulation. So we can see this really impressive, consistent decrease in GABA after, N15 and N24. And this is what's happening with ACC. Nothing. No change. So we can look at the data as much as we can in all angles. There is just nothing there. We were thinking about maybe classifying in responders, non-responders, but that doesn't really convince us. The variance is also the same. We look at changes at the local level, couldn't find any difference. So yeah, it looks like those two brain regions for the same protocol are behaving very differently.
Yeah, so Kim and I are really getting into cell type specificity, as we've told you. Can you conjecture a little bit on what differences between these two brain regions might explain this in terms of like GABAergic activity? I guess you showed us before that the baseline bold signal is much higher in the PCC.
Yeah, so is it just a state dependency sort of issue that the PCC is active?
Oh yeah.
That's what I think. That's where I'm putting all my money on, but this is just speculation. I believe that what we're observing is state dependency of TUS. TUS is showing an effect because the brain region is active, is engaged, which is, I think we discussed this, but what your colleague at Stanford is showing, Mary Maduk, I really love her work. I think it's amazing that you could see with the exact same protocol, depending on the regime of the neurons, an excitatory or an inhibitory response. So I mean, I think that's quite fascinating, and perhaps there is something there as well. But this is just one possibility, right? There is many other things. The AACC is a lot more variable across participants. Some people don't have you see this gyrus here, the cingulate gyrus. Some people have a paracingulate gyrus, some don't. So the ACC is very variable across participants, and maybe we're just sonicating at a weird place of the brain where a lot of people were just very different.
Can you get the activity to be as high as the PCC in that brain region? Like, is there, I think you were saying, how do we fire this brain region up while they're inside this monstrous MRI scanner?
Well, you can engage people in counterfactual thinking, counterfactual decision-making. So that's what I'm hoping to do next. You can do something a lot more like physiologically, lower level, like inducing
tickle their feet and then ask them if they like
No, we're doing this with a pain neuroscientist at the University of Exeter, who is more interested in like pain sensitivity and how can we maybe propose treatment for people that have chronic pain disorders.
And
but
what's
I'm
a counterfactual like question that you would ask them while you're causing pain or whatever it is you're doing?
Well, yeah, a double. No, but if you're inducing pain, just that the ACC should be active. It's the main hub of the pain network of nociception. So that's what I'm trying to do now, is to induce pain and then put them in an MRI scanner and redo the same. Yeah, so just to complete the picture, this is the neurochemistry, so the concentration of GABA within the brain regions, but we also looked at network connectivity. So this is just, I should have presented it in a different way, sorry. But if you're looking at just this brain region, this network here, this is the DMN. The DMN, when you are just resting and mind wandering at sham. And this is what's happening with TUS. It's just a lot more engaged. So this is just a coupling between those different brain regions and the DMN network. So I'm not going to go too much into detail because this is a independent, an ICA analysis, so independent component analysis. So you have to do dual regression and so on. But basically, we just see more region being active, being engaged with within the DMN after TUS. And if you're looking now at the relationship between the PCC and the rest of the brain, so you take your seed around here, and this is a misrepresentation because actually we took the focal volume from our simulation for each participant, and then we look at how this part of the brain was co-varying with a bunch of other brain regions. So this is what's happening after 15 minutes. We have an increase in coupling between the TUS and the sham. And this is what's happening at 45 minutes. So it's even more coupled with even more brain regions. So whatever we see in the network is actually more stronger at 45 minutes than it is at 15 minutes after a stimulation. And to complete the story, there was no significant change for the ACC at all. Yeah.
But if it increases at 45 minutes, did you keep scanning to see when it decreases again?
No.
Missed opportunities. We will never forget them. Elsa, how do we read this plot on the right? I see these all the time from you and from Leonard, and I never really know what I'm looking at. Can you give us our listeners a quick micro lecture? What is this spider looking thing?
So each line here is the relationship between so this is just the PCC relationship with other brain regions. So say this line here is to what extent the PCC co-vary with the putamen on the right hemisphere. This line here would be to what extent it co-vary with the putamen on the left hemisphere, and so on. So you have this yellow line here, which is the baseline, like the sham condition. So some brain regions, actually, it's not much co-varying at all. Look, it's around 0. There isn't much relationship at all between these two brain regions. Whereas for some others, you know, these guys, for example, the superior frontal gyrus, big, strong correlation to, you know, at rest. But then after TUS, you can see that this has changed. So again, the run 1 is 15 minutes after sonication. Run 2 is 45 minutes after sonication. So for most brain region, I've just presented a few, and we wanted to be comprehensive with this plot. So we didn't want to sample just, you know, the brain regions that show an increase, but a lot of different brain regions to show that it's not true that it's just more excited and more coupled, because for some brain regions, it's actually less coupled here, right? And for some, there's no difference. So it's just interesting to see that there is a change in relationship between the PCC and the rest of the brain, but it can go both ways. It can go both, like more coupling or less.
Yeah, this is really nice. I think your work also supports the idea that TUS can be something where you expose yourself to it before doing some sort of task or something, and that the treatment may last, to Kim's point, potentially more than 45 minutes. But let's forget about those missed opportunities. Maybe on the next site.
Yeah.
I'm glad you even did 45. I mean, were you expecting it to reverse and to be nothing?
Yeah.
You did it?
We were really hoping to see, you know, like a big a deep, I mean, a big increase and then something going down.
Yeah.
So that's what Robert Chen has found with MEP. He found like an increase going up to 30 minutes and then a decrease. And after 90 minutes in some of his study, this is when things are starting to be back to baseline. But we can't really put somebody in a scanner for that long. That would be a bit unethical. Yeah.
But you could take some number of subjects and not use the early time point, but just put them in for the later time point. Or you could put them in for the early, let them rest, and then go back in.
Yeah, no, I think we definitely could do that. And perhaps we should, because we keep on saying that it's transient and it's safe, it's reversible, it's transient, but we need to prove it. I mean, I have no doubt that it's going back to baseline, but we just need to prove it.
Or I mean, I guess the next question is, when are we gonna see the next release? Are you guys doing behavior with this? Are you following up? It's really exciting, by the way.
Yeah, we have very cool behavioral.
Oh, you already
research.
have it. Oh, wow. It's not in this presentation, right? The people will have to wait.
So you did the 3 sessions, and presumably in some subjects, the sham was session 2 or 3, in which case you would show that for those subjects that it had gone back after the sonication.
Yeah, that's true.
It's weeks later, though.
That's true. Yes, yes, right.
Does it go back to like what it was weeks before? Is the resting mode network that stable? I would think the baseline would have shifted. Or
the retest reliability with sham with Gaba and and fMRI is pretty reliable. The DMN is pretty reliable across the.
That could be very interesting then.
But you're right. Some of these, we can reorder this and show those ones where sham was acquired at the end. Yeah. Cool. Yeah, I think that's it really. So in conclusion, what we've seen is that there was evidence for with an excitatory, say, protocol, although I really don't like talking about protocol being excitatory or inhibitory, because I don't really think that I don't think that it's that simple. But what we've seen is a decrease in GABA and higher coupling in a lot of region. So it does suggest that there is an excitatory effect of TUS in deep cortical regions. Now we don't know why there is this big difference between ACC and PCC. Is it because it's state dependent? Is it because, as I said, the ACC is very variable across participants? Is it because the tissue is very different for these 2 brain regions? And then there was something else that struck me this afternoon for some reason. Maybe the protocol works well for some region. It might be excitatory for some region, but maybe not for other regions. So it might also just be that the same protocol. Well, yeah, it's kind of the same thing, but I'm thinking a little bit more about the protocol that we're using and why we're seeing what we're seeing. Anyway,
it's so easy to want to ascribe what you see or differences in what you see to the protocol, when I think you just have shown us so clearly that there could be state dependence, as well as the types of the cells that are there, and then after we kind of disambiguate those, maybe think about the protocol. I don't know, it's just my opinion, but it's you've shown us just a beautiful example here of that.
Yeah, thank you. Yeah, I think it's very exciting. It doesn't answer a lot. It opens a lot more questions than I think it addresses, but it's still very exciting because we do have evidence that we can induce effects that are quite long lasting. Now we can, open the question on what is it exactly that we're doing? Is it long-term potentiation? Is it things that if you were to repeat it over a course of a treatment, say weeks, could that induce like really, really long lasting effects? That's what you want for treatment, right? all of these questions. And is it enough to induce behavioral effect? You'll know this at 123.
Oh, wow. We got to wait a while for that.
All right, excellent. Yeah. So we've had a great time talking to you throughout all these slides. I don't know, do we have any last minute questions there, Keith, before we wrap up?
Got through most of my questions. TUS versus FUS. Why are you calling it TUS? I think there's a war brewing. Kim and I, we're very FUS. We like to call it focused ultrasound.
No, I'm on board with TUS.
Oh, you are? Oh,
absolutely. Man. It's parallel
no.
to TMS and TES and TDCS to say TUS.
I'm a lone ranger.
Actually, in Oxford, we called it fun. And I still think that neuromodulation is better than stimulation. But that's
I agree with that. I'm more for fun than I am for TUS. But I'm still a FUS guy, so FUS forever. Oh, actually, one more question, if people are still listening, if anyone was listening. Our viewership's been up, I think, but hopefully this lecture gets them on board. When you measure with fMRI, do you see increases in blood flow to that region? Because you showed a decrease in local GABA concentrations. So is that another metric you're going to have? Or did you monitor that? Sorry, I got a little twisted with that.
Oh, you're talking about blood flow. So a lot of people look at ASL, arterial spin labeling, and I guess that's more not blood flow, but looking at blood volume. So, but you haven't done that, Elsa.
No, we haven't, but I think it would be very cool.
I'll take bold too. Well, I guess I was thinking blood volume maybe instead of flow.
Oxygen level dependent, yeah. But bold, so I mean, decrease in GABA, again, that means excitatory, right? We can talk about what we think is happening, but it means that overall there is more excitability. What's the correct word? It's more exciting.
Did you see more bold signal in the PCC as a result?
So I've spent a lot of time looking at the local, at bold, at the local level in the MRS. I looked at the spectrum, I looked at autocorrelation of the data. There wasn't actually much at all, even for the PCC, because I was hoping to see something at the ACC level, because, you know, I was quite obsessed, like maybe there's something that we're missing with GABA, but it still should show something. But no, I couldn't find anything. But yeah, the most surprising thing was that even the PCC with itself didn't seem to be that much more connected with itself.
I'm kind of wondering if monitoring GABA with this technique is better resolution than bold stuff. Not that I know much about either one of these. But I know they're both, they're different and they have inverse correlations to each other from the review that you made us read. I read this.
Yeah, we didn't find that though. It was
maybe GABA's the way to go then, Elsa. I think you've struck upon a really nice measure of what's going on from TUS or fun.
Yeah, I think it's definitely interesting. Then there is the problem of what are we really measuring with GABA MRS, right?
Mm-hmm.
It's for another lecture, I think.
Yeah, we need another lecture on that. Well, thank you so much, Elsa. This is awesome.
Well, thank you for having me.